Good afternoon, students. So, we'll carry on with this chapter today, that is Principles of Inheritance and Variation. And today we are going to start with linkage and recombination. Now, what do you understand by linkage? Linkage is the close association of genes when we are talking about genes or other DNA sequences on the same chromosome. The closer two genes are to each other on the same chromosome, the greater is the probability that they will be inherited together. Have you understood anything? Let me explain you with this chromosome. How we say that they are linked or they are not linked. Now in the first diagram you see that in a chromosome two genes are present, gene 1 and gene 2, and their distance between the two genes is very small. So this means they are linked. During crossing over and meiosis they will be carried over. The more chances will be that they will be carried over together in the same chromosome. Now, if we talk about two different chromosomes as shown in the second diagram, you can see the of two chromosomes. Gene 1 is present on one chromosome and gene 2 is present on the second chromosome. Here, they are not linked. Then there is one more case, that is the third diagram. On the same chromosome, if the gene 1 and 2 are present distantly apart, you can see here that the distance between the two is great enough that they can be inherited independently, and during crossing over they can go to different paths, that is recombination can occur. We will come to this term recombination also. So it's clear. So in the first diagram the genes, they are supposed to be linked because the more tendency is to be inherited together. In the second one they are not linked, and in the third one also they are not linked. Okay. So why genetic linkage? We are talking about genetic linkage. So why this genetic linkage? Because in organisms genes are far more in number than the chromosomes. This means that each chromosome contains many loci for different genes. Now here the data is given for humans, which has 23 pairs of 46 chromosomes, and the inheritance is something like this. You can see in this diagram that this part, if we open of the chromosome, if this part is unfolded, you can see the DNA, and a part of the DNA here on top shows gene 1, and then the lower part carries on the gene 2. So if these genes are close together, more tendency is that they will be inherited together. That is what we mean by the term linkage. Now we come to the next term, that is recombination. So recombination is a process by which pieces of DNA are broken and recombined to produce new combinations of alleles. This recombination process creates genetic diversity at the level of genes that reflects differences in the DNA sequences of different organisms. So how do we explain it? We will take the help of this diagram. Here you see presence of a homologous pair of chromosomes. Both the homologues are represented by two different colors, and the genes capital A, small b are present on one of the homologues, and small a and capital B are present on the other homologous chromosome of the same pair. Now this is one of them is considered as the maternal and other is as paternal. So this is one set of homologous pair of chromosomes in a diploid organism. Next, you see crossing over is taking place. That means during meiosis, the crossing over is taking place. And then finally, these recombinants are formed. That is how they are inherited, parental types as well as the recombinants. Here you see they are formed in equal proportion. They are supposed to be formed in equal proportion also if they are present at a distance from each other. So you can see the distance between the two A and B is great enough to be inherited independently. That is independent assortment. So after meiosis, you can see in the haploid gametes, the inheritance will be something like this. Like in the first, it is shown the parental combination, that is capital A with small b. In the second is the recombinant is formed, that is capital A with capital B. In the third chromosome is small a and small b, that is again a recombinant, and again a parental type, that is small a and capital B. So this is how it is inherited. Right? Clear? This is going to help you understand what we are going to discuss. Now actually you see this is also a similar diagram showing homologous pair with two different colors, and after the crossing over, the resultant should be something like this as shown here. A small segment showing the orange color on the green one, and on the other this segment has been shifted to the other homologous during crossing over. Right? That is how capital A, capital B, and then small a, capital B, or capital A, small b, that is the recombinants are formed. But in actual fact, it is seen sometimes that the expectant result is not there. That is, we expect the recombinant chromosomes to form, but they are not being formed very often. So this means the expectant results will deviate from the normal Mendelian ratio. Right? This is what we are going to study today. So if we take both these terms together, linkage and recombination. So what is it? This refers to the association and co-inheritance of two DNA segments because they reside close together on the same chromosome. This is very important, close together on the same chromosome. So recombination is the process by which they become separated during crossing over, which occurs during meiosis. I hope now it is clear to you what we understand by these two terms. If it is not, let me tell you again, and you are going to tell me the answer here. So gene linkage and recombination. Here we take two genes as capital E, capital M, dominant, and small e and small m as two genes which are recessive. Now these genes are far apart such that crossing over between the alleles occurs occasionally. Right? The distance between the two is so much that crossing over can occur between the alleles occasionally. So now you have to give me the answer that which statement is true of the gametes. All of the gametes will be capital E small m and small e capital M, or there will be equal number of all these, or there will be approximately equal number of capital E capital M and small e capital M gametes, or there will be more capital E small m gametes than the small e and small m gametes. Right? So regular gametes will be capital E small m, small e and capital M as shown in the parental diagram. So regular gametes are in majority, whereas recombinant gametes, they are small in number. That is, they are capital E, capital M, small e and small m. These are the recombinants here. But their proportion is varying, and this proportion varies according to the distance between the two terms. Now before we take up this, I would like to make few things more clear to you. Now, like the difference in the behavior of the unlinked and linked genes in their assortment during gametogenesis, right? So unlinked genes, what are they? The genes located in different chromosomes. This is very important. In different chromosomes, they assort independently and they give the Mendelian dihybrid ratio, that is 9:3:3:1, and they give a test cross ratio of 1:1:1:1, right? This we have studied earlier, right? So here the four types of individuals in the dihybrid test cross progeny, they are found in equal proportion, that is 25% each. And of the four, two are parental types and the other two are recombinant types. And parental and recombinant frequencies are often they are equal, that is 50% each. Now what happens in the linked genes? This was about the unlinked genes. Now we'll talk about the linked genes. Now the genes located in the same chromosome usually do not assort independently but remain together in the gametes and the offspring, give a dihybrid ratio of 3:1 and show a test cross ratio of 1:1. Now you see the difference between the two. So the progeny here comprises in the test cross only two types of individuals, they are which are produced in equal proportion, that is 1:1. Now both are parental forms and no recombinants have been formed, they are formed. Now this is a deviation in the dihybrid test cross ratio, that is 1:1:1:1. We have only 1:1. Now this is an evidence of gene linkage. Clear? Unlinked genes, they show direct Mendelian ratios, but the linked genes, they are deviated from it. Now as far as how this linkage it was discovered, the principle of linkage was first discovered by Batson and Puneet in 1906 in sweet pea, that is Lathyrus odoratus. But it was put forth as a regular concept by Morgan in 1910 from his work on fruit fly, that is Drosophila melanogaster. Now as far as the chromosome theory of linkage is concerned, according to Morgan and Castle, who gave this chromosome theory of linkage, the postulates of it are: genes lie in a linear order in the chromosomes. The genes which show linkage, that is stay together in hereditary transmission, are located in the same chromosome. Genes tend to stay in parental combination except for occasional crossing over. The distance between the linked genes in the chromosome determines the strength of linkage. The closely located genes show stronger linkage than the distant genes because the latter are more likely to undergo crossing over than the first case when they are present close together. I hope I made it clear to you. I am putting more stress on what is linkage and what is recombination. Before you understand what you have to study, that is the Morgan's dihybrid cross showing linkage and recombination. So, going further or talking in depth about linkage, we can say that linkage is mainly of two types, that is complete linkage and incomplete linkage. Now, what do we understand by these terms? When the genes are closely located in the chromosome, they show complete linkage as they have no chance of separating by crossing over, and they are always transmitted together to the same gamete and the same offspring. So the parental combination of the trait is inherited as such by the young one. This is what is complete linkage. No recombinants. In complete linkage, no recombinants. And for this we can discuss the example of Drosophila melanogaster for its wild type and the mutant type for the inheritance of eyes and the wings. We'll take it up. Now the next type is the incomplete linkage. Now what is that? When the genes distantly located in the chromosome show incomplete linkage because they have a chance of separation by crossing over and of going into different gametes and offsprings. So here you understand the difference. In complete linkage, there is no recombination. In incomplete linkage, it depends on the distance between the genes because there are some chances of crossing over, and during crossing over, formation of recombinants. Now, an early case of incomplete linkage which allows the separation of linked genes, it was noted by Batson and Puneet in sweet pea, that is Lathyrus odoratus, in 1906, as I told you earlier. Clear? Now we will talk about another term, that is linkage groups. So what is a linkage group? All the genes in a particular chromosome form a linkage group. Clear? All the genes in a particular chromosome form a linkage group. Now the genes in a particular chromosome, they have their alleles in the corresponding homologous chromosome also. So a pair of homologous chromosomes are said to form a single linkage group. So the genes occur in as many linkage groups as the number of the pair of homologous chromosomes. This also means that the number of linkage groups in a species corresponds to its haploid number of chromosomes. This is what is known as limitation of linkage groups. The size of the linkage group depends on the size of the chromosome. So this means that small chromosomes have small linkage groups and large chromosomes have large linkage groups. Drosophila melanogaster has 4 linkage groups. 3 large, 1 small. which correspond to the 4 pairs of chromosomes. Now there is one more thing which is known as sex linkage. Chromosomes, they are autosomes and sex chromosomes. So we are talking about the sex linkage. The sex linkage means that the linkage between on the chromosomes X and Y, the sex chromosomes. So the sex chromosomes X and Y, besides carrying the sex determining genes, they also bear gene for some other characters. Now these additional genes which travel along with the sex controlling genes from one generation to another are known as sex linked genes. The characters controlled by the sex linked genes are termed sex linked characters. Inheritance of sex linked genes is known as sex linked inheritance, and the location in the sex chromosome is known as sex linkage. Most sex linked genes, they are located on X chromosome called X linkage. A few genes occur on the Y chromosome and they form Y linkage. The Y linked traits are transmitted only through the male. For example, gene for sex determination in mammals, the Y chromosome carries very few genes, right? Now the sex linkage was first discovered by Morgan again in 1910 in the fruit fly Drosophila melanogaster. Fruit fly has XX and XY sex chromosomes in the female and male. Like humans, it has XX as the female and XY as the male. Its eye color is sex linked. Allele for the eye color gene is located in the X chromosome. So this means there is no corresponding allele in the Y chromosome. All this is very important, so please listen attentively, because when I am going to discuss this cross, then you will understand all these things. Why I am taking up all the terms and clarifying them. The male expresses a sex linked recessive trait even if it has a single gene for it. Why? Because there is only one X chromosome in male, whereas the female expresses the recessive trait if it has it on both the X chromosomes. So the normal eye color for Drosophila is red, and it is dominant over the mutant white eye color, right? Remember this, okay? So now we will discuss this in the cross. Now make one more thing clear. Here you see dihybrid cross which was conducted by Morgan. Now here you see the meiosis with no crossing over between the genes and meiosis with a crossover. So here the two genes, you see the parental types capital AB and small ab. Without crossing over, all the parental types are produced as a result of meiosis, and in the second part there are two parental types and two recombinants. So in case meiosis during the crossing over occurs during meiosis, then the recombinants are formed, and if there is no crossing over during meiosis in this particular part, then only parental types or the parental type of combinations, they are formed. So whatever is combination is present in either of the parent, that is known as the parental combination in the offspring, and if there is a change in the character, one from one parent and the other from the other parent, then such a characteristic combination is known as a recombinant. Clear? Okay. So now we'll talk here about the cross between the yellow-bodied white-eyed female and brown-bodied red-eyed male. Please focus on one more thing, that is the distance between the two genes. The yellow-bodied and white-eyed gene, they lies very close to one another, and here you see in the female it is present on both the X chromosomes, but in male, all because it has only one X chromosome, so this characteristic is present only on one chromosome, that is the X chromosome, so it expresses the same character. This is the parental character what we are going to discuss. Okay, so you have seen it, this parental characters. Now we also call this male as the wild type. Now in the F1 progeny, obviously the X chromosome from the male is going to be inherited by all the females. So all the females, they become brown bodied and red eyed, that is the wild type, because it is a dominant character, whereas all the males, they show yellow body and white eye, because only one X chromosome is present, although it is the recessive trait, but only a single X chromosome is present, so it expresses itself. Then we come down to the F2 generation. So the gametes are formed and we reach to the F2 generation. So here you see we don't find the expectant result of 9:3:3:1. Here the parental combinations are 98.7%, whereas recombinant types are only 1.3%. So on breeding, these breeding experiments, they show this result. So why the expectant ratio was not achieved? Because of linkage. So here they are closely linked together. Is it clear? If you have any doubts, you can text me. I'll clarify the doubts. Now I'll explain you with 2-3 examples the same thing. So here you see they have in this, we see in the actual Drosophila drawn here. So in the parental generation, which is wild type, we are taking two characters, that is the gray body with the normal wings, and double mutant type, that is black with the vestigial wings, that is rudimentary, very small wings. So two characters we are taking, right? Now F1, which is formed, here we perform the test cross with the F1. So F1 hybrid with gray and normal wings, gray body and the normal wings, is crossed back with the double mutant parent, that is the test cross, and the expectant offsprings, expected, right, the expected offsprings, that is because of independent assortment which we expect as the parental and the recombinant types, are these four. That is, one is the wild type, then black body with vestigial wings, then gray body with vestigial wings, and black with normal wings. Now the first two, that is the wild type, gray body with normal wings and black with vestigial wings, they are the parental phenotypes, whereas the gray vestigial and the black normal wings, they are the recombinant types. Expectant is the proportion of independent assortment, 9:3:3:1, but we will not achieve it, so I'll show it to you. But before that, I will discuss another, this character 1 monohybrid cross with you. Here we see white-eyed female with a red-eyed male. Remember, the male has only one X chromosome, so the character which is present on it is going to express itself. So in the resultant we find all the females, they are having red eye. That means the red was dominant, whereas all the males, they have white eye. This is the F1 progeny in a single inheritance, monohybrid. Right? This is the monohybrid cross, but the proportion is not so. So this offspring here is all females, they are red eyed, whereas all male offsprings, they are white eyed. Now you can tell me why? Because the red eye color is dominant character, and in single dose, that is this on present on single chromosome, X chromosome, it can express itself, whereas in the males there is only one X chromosome. So whether it is red or white, it will express its color. And the white eye X chromosome, this is received from the mother. Right? So we come back to the dihybrid cross which we were discussing before. Right? Grey body with the normal wings and the black one with the vestigial wings. So after the test cross, the resultant was formed there, right? But we did not discuss about the proportion. So genes on different chromosomes, 1:1:1:1. So genes on the same chromosome, and there is no recombination, that is 1:1. Now genes on the same chromosome and recombination is not expectant ratio. You see the number here. First 965, then 944, 206, 185. So this is not 9331 ratio. There is difference in this ratio. And this difference from the regular Mendelian ratio is due to linkage. Now the difference in the linkage is also dependent on the distance between the two characters which we are talking about. Less the distance between the two, more is the linkage, and more the distance between the two, less is the linkage. So Morgan's student Alfred Stuyvesant, he used the frequency of recombination between the gene pairs on the same chromosome as a measure of the distance between the genes and mapped their position on the chromosome accordingly. Do you get it what I mean? From this linkage and the percentage of linkage, he developed a system to measure the distance between the two genes on the chromosome because they are present on the same chromosome. So percentage of linkage and recombination decides the distance between the two, and this is what is known as mapping of the chromosome. You know, today genetic maps, they are used extensively for sequencing of the whole genome, right? You know, whole genome sequencing is done in humans also under the Human Genome Project, which was a mega project. We'll discuss it later on. So this is how the linkage affects, or the distance between the two genes, you can say, affects the inheritance for linkage or for recombination. Now we come down to the next topic of today's lecture, that is sex determination. The sex determination system is a biological system that determines the development of sexual characteristics in an organism. Most organisms that create their offspring using sexual reproduction have two sexes. Occasionally, there are hermaphrodites also, but usually there are distinct sexes which we normally call as male and the female. Now we are going to discuss the inheritance of this sex in humans, in birds, in insects, right? Please look at this diagram. So we'll take up the example of grasshopper first, right? We'll start from there. So grasshopper is an example of X0 type of sex determination in which the males have only one X chromosome besides the autosomes, whereas the females they have one pair of X chromosomes, right? But a number of other insects and mammals, including man, the XY type of sex determination is seen where both male and female have the same number of chromosomes. So among the males, an X chromosome is present, but its counterpart is distinctly smaller and is known as Y chromosome. Females, however, have a pair of X chromosomes. Both males and females, they bear same number of autosomes. For example, in humans, we have 23 pairs, of which 22 are autosomes and one is the sex chromosomes. XX in the females and XY in the males. In Drosophila also, the males have one X and one Y chromosome, whereas the females they have a pair of X chromosomes. Now what we are talking about is besides the autosomes, of course. Now we have discussed the X0 type and XY type. But in both these cases, you see the males they produce two different types of gametes, either with or without the X chromosome. Okay. So that means either they have like in the grasshopper, either with X chromosome or without it, or in the case of humans or Drosophila, gametes with X chromosome and gametes with Y chromosome. So such type of sex determination mechanism is known as male heterogamety. Now in some other organisms like birds, a different mechanism of sex determination is present. In this case, the total number of chromosomes is same in both male and females, but two different types of gametes is present in the females, that is female heterogamety. So in order to have a distinction with the mechanism of sex determination of XY, the two different sex chromosomes of a female bird have been designated as Z and W. In these, the females they have one Z and one W chromosome, whereas the males they have a pair of Z chromosomes besides the autosomes, of course. Right? So this is about different organisms, the inheritance of the sex chromosomes. And let me tell you one more thing, that during the spermatogenesis and oogenesis, there is one difference. During spermatogenesis, you see 4 gametes which are formed are all functional. So in heterogamety of male, the 50% of the total sperms produced, they carry X chromosome and 50% carry the Y chromosomes besides the autosomes. But the females, they produce only one type of ovum with the X chromosome. But there is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. Now, if the ovum fertilizes with a sperm carrying X chromosome, then the zygote will definitely develop into a female. And if it fertilizes with the Y carrying chromosome of the sperm, then it will result into a male offspring. Right? So, it is evident from the genetic makeup of the sperm that it will determine the sex of the child. And moreover, in every pregnancy, there is 50% probability of either a male or a female child. But in our society, you know that females are considered for the determination of the male or the female child, but now you know that this is only a myth. Actually, the male shows the male heterogamety. It produces 2 types of gametes, X carrying and the Y carrying gametes in equal proportion, and there is equal probability of either one fusing during fertilization, and that is what is going to determine the sex of the newborn. Clear? Thank you, children. Stay safe, stay healthy.